και να περάσουμε σε άλλη μια πολύ ενδιαφέρουσα παρουσίαση.
Γεια σας, μ' ακούτε? Λέγομαι Γιώργος Βασματζής, δουλεύω στο MAYO Clinic. Αυτή τη στιγμή είμαι όμως στα Μέθανα, σε ιστιοπλοϊκό, και σας ευχαριστώ πάρα πολύ για την πρόσκληση. Αν δεν σας πειράζει θα το γυρίσω στα αγγλικά, γιατί αυτού του είδους τα talks δεν μπορώ να τα κάνω στα ελληνικά. Θα χρησιμοποιούσα πολλά ελληνικά-αγγλικά και θα ήταν λίγο δύσκολο. Οπότε, sorry for this, so I'll be speaking in English. Evie asked me to talk about precision oncology and the digitization that happens lately. But I'll be talking mostly about functional genomics, which they do use intelligent systems, but not necessarily artificial intelligence, but more mechanistic intelligence. And I'll explain what I mean by that. The functional genomics field is an older field, but I think the functional genomics and systems biology is becoming more of a reality lately than it was about 10 years ago when it was conceived. Here are my disclosures. I've been at MAYO Clinic for 21 years. I'm an electrical engineer actually by training way back 30 years ago, but slowly I moved into genomics and information, and I also direct a laboratory, an experimental laboratory at MAYO, the Biomarker Discovery Laboratory. So I have both engineering, experimental, as well as theoretical skills, and I also have a few disclosures. As you know, my goal at MAYO Clinic for the last 21 years that I've been there is to bring genomics to the clinical practice. And in the beginning, people didn't know what genomics are. It was an extremely difficult task, but I think right now we're getting very close to bringing it to the practice. But what is really the challenges in individualized medicine in oncology, especially for advanced cancers, is that they are extremely complex genomically. If you look at cancers with multi-omic techniques, on the DNA level, the RNA level, the epigenetic level, you will see a lot of things happening at the same time. And also most of those things, actually, we don't really know what they mean. We don't know the function of the genes that they are being hit or how they interact with other genes for many of those. We do know some, maybe 30, 40%, but not all of it. So there is really a knowledge gap. But there are other issues. There are more technical issues on how you acquire the right tissue, especially in advanced cancer, where you don't have surgical tissue. You have to get the right biopsy. You need interventional radiology. There are issues with tissues with cellularity of the tumors that we get, and then how do you validate what you see with the genomics? And also, how do you apply that when there is a standard of care, and especially in the United States, which is very difficult to go against? There are a lot of barriers, regulatory barriers that we have to go through. So there are all these challenges, and but with respect to complexity, I wanted to dwell on that a little bit more. Here, I'm showing you the genome plots of 6 different gliomas. I'll explain what those lines are and what these bubbles are eventually, but just by looking at the genomic complexity of those 6, you can see they are very different from one another. So what are all these lines here? Well, the way we try to visualize the human genome is by this, we call it the Genome U Plot. It has all the chromosomes shown in the 2 dimensions rather than in a circle, which is a circus plot. This is the more like a linear way spreading the genome into 2 dimensions. And on the left side, you have the larger chromosomes. On the right side, you have the smaller chromosomes. And if you see a line like this magenta line that goes between chromosome 2 and chromosome 8, this is because we've seen sequences of by using next-generation sequencing, we see sequences that span between those 2 spots in the genome. Each one of those dots here, the gray dots, represent about a 30 kb window. And the higher the dot, the more sequences you get in that window. Okay, so when you see gray, that means that you have the normal or 2N genome, the 2-copy genome. But the red dots, as you can see here, are lower. And the reason they are lower is because you have less sequencing in there, which indicates a deletion. And of course, sometimes you have the sequences that span those deletions. So you have two different types of information to figure out that you have a deletion. Both the copy number, as we call it, that's going down, as well as the lines of the sequences that span between those points. And you can see that sometimes those are very complex. This is a chromotriptic event, creating all those deletions, but in a very complex manner. Incidentally, up here on chromosome 21, there is a deletion, and that's the deletion of the ERG, TMPRSS2, brings TMPRSS2 and ERG together, and that's a typical genomic deletion in prostate cancer. It hasn't been found anywhere else, as far as I know. Now, going back to the figure I showed you earlier, here you see 6 different gliomas. All those different lines here would be translocations. This is a chromoplectic event, showing these 3 different regions. This is a chromotriptic event. This is another chromoplectic event. All those bubbles here are mutations in this case. There is chromoplexis, but there is a lot of mutations. This is a hypermutated phenotype. Here is another glioma that has a very sharp amplification on chromosome 7. So chromosome 7 is blue, that means it's elevated, it's 3N, but it has this sharp amplification, which is around the EGFR, okay? And then there are other things that are happening as well in these different gliomas. A very typical thing that you see is something happening on chromosome 9, most of the times that hits P16. So what is functional genomics? As I was saying before, it's not exactly artificial intelligence. It is more trying to understand the function of things. And one of the ways that we try to understand functions of such a complexity is by visualizing it first. I mean, the idea was, okay, let's see how that complexity looks. And we needed to find ways to visualize that complexity. So we developed this genome plot up here and something what we call the pathway plot. On the other side of the functional genomics, we have a functional validation method. This is developed by another Greek, Panos Anastasiadis, who is in the Florida campus at MAYO. And he has developed this hang and drop method where we take tissue from patients and we grow it in small spheroids, and then we drop it on a on the dish. Then you have a 96-well format, actually, in this case. So you do a lot of experiments here, and then we put drugs, and I'll explain that a little bit more. So this whole functional genomics is being applied in a project we call Xvivo, which is supposed to bridge the discovery science with clinical practice. And you're supposed to go from pathways and microenvironment and multi-omics technologies to, let's say, oncology. And that's the project. So combining state-of-the-art genomics with 3D microscans, drug sensitivity screens to predict the most effective treatment for each individual patient, right? So it combines both a theoretical and experimental way. Typically, on a patient, we take, let's say, some brain tissue. We split it into two. Part of it goes frozen for sequencing and analysis, and we use a group called Think Tank, a tumor board, to analyze the data, and then we go in this validation model. And sometimes, when we find something effective, sometimes we're able to go to the patient. But that requires a CLIA test to corroborate the evidence. We've done actually close to 200 cases right now, spanning many different diseases. And we have engaged a lot of oncologists in this effort. And we found that has actually opened their eyes in some ways, because oncologists really didn't have full genomics to see what is going on. Sometimes they see patients, they see symptoms, they see things, but they don't see the genomic complexity behind the tumor. So what is our core engine? As I said, first, an oncologist will see a patient. They think that they don't know what to do at that time because they've tried different things that didn't work. We will take a piece of tissue and we'll split it in two ways. We'll go fresh and we'll do the cancer biology. We'll do the 3D microcancers the cryopreserved tissue. And on the other side, we do the frozen tissue, the pathology, and we do genomics. All this information comes together to the think tank, and then we create an integrated report and we go back to oncology. Okay. That process requires a lot of different disciplines together, and it is really more like an engine, okay, where you have the engine in the middle with all these different pieces and all these different people that they work together, and the input of the engine is clinical questions, and the output could be precision medicine. So in order to get that, you need multiple different capabilities, as I said, working together. One is the tissue sample collection and storage. You need to have pathologists, anatomic pathologists that will do the not only the tissue acquisition, but figure out what kind of tumors we are looking at. We also have a lot of dashboards and visualization techniques and bioinformatics to understand what is going on in the genomic part. And we have the validation optimized 3D culture method to validate what we are observing. And we also have monitoring ways with cell-free DNA. This is our dashboard. Every patient has a dashboard like this, and you can follow the patient through those dashboards by clicking all these different spots and visualizing data. Or, for example, in this case, you can click the Genome Explorer here, and it will take you to something like this. And that's a Genome Explorer of a patient. The bubbles here are mutations. The spikes here are amplifications. The magenta lines are translocations. The cyan lines are fusions. And you can actually use it as a microscope pretty much and go to places like, for example, here we're zooming into ERBB2. That's where HER2 is, and the up and down now is expression. So this is an amplified area of the genome, and that increases the expression on HER2. And the more you zoom in, the more information you get. Okay. Now, of course, this information, this is overexpressed compared to what is going on in other cases. Now, the other visualization that we use is the Pathway Explorer. Here, all the alterations are being mapped on the pathways. Like, for example, you can click here and you will see that in this case, FGF18 has a mutation. The boxes, the genes here are color-coded according to expression. So you can go and look at different genes and figure out what genes are being hit in all these different pathways. So I don't have too much time to explain all the details of these techniques, but of course, the idea here is to make it easy for everybody to understand what is going on. So we use all this information, we look at the visualization, and then we have genomicists that would use algorithms to extract the right information. And then we have many techniques, both algorithmic and experimental, to find functional, to integrate the analysis and find functional things on the genes, like activated pathways, fusions, or even figure out what is happening with the immune system, because we have all this information together. So now here's a case, for example, it is a glioma case. It has a gain on chromosome 7. It's got an amplification right here. Guess what? This is EGFR. By looking at that amplification, we know this amplification is happening with double minutes. Right here on P16, we have 2 steps. That means P16, which is on chromosome 9 right here, has been double hit. There's a chromoplexis here, a lot of other things. And then you can also zoom into the areas, as I showed you. And in this case, P10 is also a double hit of P10. One allele is mutated, the other one is deleted. We can see all this information pretty easily. And then, as I said, we can map this information on the pathways. And not only that, we can also solve kill-the errors with the functional genomics. So in the functional side, we take tissue, but now we cryopreserve it. We keep it live. And as I said, we use this hang and drop model where we make the spheroids that they drop on the dish, and then we can do a drug screen, and we have kill curves for different drugs. These drugs are usually being found by the functional, the comprehensive genomic step that I showed you earlier, and sometimes we can go to the patient. The 3D microcancers that Panos has developed is a non-proliferative model because it tries to recapitulate as well as it can the actual tumor of the patient, preserves microenvironment, preserves heterogeneity, and it even works with immunotherapy because it preserves T cells and others, and it's very quick and inexpensive. He's able to have about a 90% take rate. It's pretty amazing, actually. And we've been able to look at both the microcancers and the original tumor and see that by genomics, it's almost identical. So he can even do combination therapies in some ways and figure out with a little bit interesting math on if things are synergizing. I don't have time to go into that either. I'm going to give you a quick example. Here's a patient we call S11. The foundation one of the patient was underwhelming in some ways. It didn't have too much information. Perhaps it saw a mutation on NF1 and suggested trametinib. Found the VUS in BB2, a bunch of other things, but really didn't tell the oncologist what they should do. When we look at the case, we see a lot more complexity. There is a gain on 1Q, there's a deletion on 1P, there is a mutation on CDH1, double hit on CDH1 actually, deletion and mutation, double hit on P53, a lot of other mutations by looking at the bubbles. And looking at the VUS at ERBB2 and looking more closely to the information, we found that that might have been an activated mutation because the expression of HER2 was pretty high. And when we tried the functional validation, a lot of the drugs against HER2 worked very well in the kill curves. And in this case, we were able to go to the patient with afatinib and show a pretty good 6-month response on that particular patient. We have a few more examples like this. This is another HER2 another breast cancer patient. And in this patient, we tried a combination of vorinostat and eribulin, and that worked very well. And it worked in the patient very well. As you can see, there was some decrease of the level of the volumes of the cancer, and gave the patient a few months that they wouldn't have otherwise with their family. And here's another breast cancer. Again, similarly, we were able to direct the therapy of that patient, and we saw a pretty good response. Here's a rhabdomyosarcoma for a little girl. Pretty complex genome again, but found a couple of hits in KMT2T and KRAS, which suggested panobinostat and trametinib that also gave that patient a few months that it wouldn't have otherwise. Again, that was found in the 3D microcancers and then corroborated in the patient. Here's a lung cancer patient of Dr. Leventakos, who is an oncologist at MAYO. In this case, there was again a HER2 amplification. It was a squamous lung cancer. They usually don't act on that, but through this, he acted on the patient, and you can see a clear response for that patient before and after pictures here. We also see sometimes that we have two different hits in driver genes. Here is a HER2 amplification, and this is an FGFR3 amplification. And we see that the synergy of the two drugs do better. We also can understand sometimes why something doesn't work. In this case, this is a melanoma. That melanoma was given immunotherapy. It was working quite well for a couple of years, but then there were some lesions that were not responding. And when we looked at the genome, we see on chromosome 15, one allele was deleted, and there was a mutation in beta-2M, pretty much knocking out the MAC presentation of that patient, which is why the immunotherapy didn't work. You can see it didn't work in the 3D microcancers as well, but there were a couple of other things that we could have tried in that patient. We didn't in this case. So what are the next steps? I kind of give you a little bit, a few examples on how we can use functional genomics. This is, complete genomics or comprehensive genomics combined with this functional validation. And what we want to do right now is to put it in the clinic. We are not able to do it yet with clinical tests because none of those tests are yet at the CLIA level, as we call it. But we are able to do it with IRBs. And as a matter of fact, we are writing an IRB right now, which is an advance from this previous IRB, and it's supposed to be able to bring this, put some of these results into the patient's medical record and allow the clinicians to act on them. So we are also developing ways to do better pathway analysis, even automate the 3D microcancer process. I'm particularly interested in the pathway analysis because, as I said, there is a knowledge gap, but using the tools and the visualization, we can do even reverse engineering and, you know, sometimes open up the black box, as it was mentioned earlier, learn something and put it in the mechanistic models. That's all I had. I'll be happy to take any questions. Thank you.
We can switch to Greek for a bit.
Absolutely.
Καταρχάς, η κυρία Χατζιανδρέου μας υποσχέθηκε πολλά και εντυπωσιακά πράγματα και you delivered. Σας ευχαριστούμε πάρα πολύ. Και φυσικά ευχαριστούμε για το γεγονός ότι εν μέσω των διακοπών σας αφιερώσατε χρόνο για να μοιραστείτε πράγματα που δουλεύετε στο MAYO Clinic. Και πάλι δεν ξέρω αν έχουμε κάποια ερώτηση από το κοινό μας.
Να ρωτήσω εγώ κάτι, Θάνο, μπορώ? Ήταν εντυπωσιακή ομιλία. Σε σειρά και των προηγούμενων εντυπωσιακών, νομίζω ότι είναι κρίμα που δεν είναι περισσότερο γνωστά όλα αυτά τα ερευνητικά και ελπίζω σύντομα και στο clinical practice πράγματα στην Ελλάδα. Με εντυπωσίασε περισσότερο απ' όλα το γιατί δεν δουλεύουν κάποια φάρμακα ή στοχευμένες θεραπευτικές προσεγγίσεις. Θα μπορούσε στο πλαίσιο της ερευνητικής εντός εισαγωγικών ανάλυσης να πάει κάποιος αντί για το best responser, το worst responser, δηλαδή να δοκιμάσει κάποιους ασθενείς που δεν πήγαν καλά ενώ έπρεπε να έχουν πάει σε ορισμένα φάρμακα με αυτά τα πρωτόκολλα έτσι ώστε να ανακαλύψει ενδεχομένως άλλες διασταυρώσεις ή μονοπάτια.
Ναι, πιστεύουμε ότι και το negative result είναι πολύ σημαντικό. Τουλάχιστον, γιατί πολλές φορές αυτό που γίνεται είναι δίνουν ένα φάρμακο μετά το άλλο, περιμένουν μερικές φορές 2-3 εβδομάδες για να δουν ότι δεν θα δουλέψει και χάνουμε χρόνο και χάνουμε χρήμα. Οπότε κάτι τέτοιο θα ήταν πολύ καλύτερο να σου δείξει ότι κάτι δεν δουλεύει, κάνε κάτι άλλο τουλάχιστον. Αλλά και αυτό ακόμη πρέπει να είναι clear test. Δηλαδή πρέπει να αποδειχτεί με κάποιο τρόπο για να μπορούμε να χρησιμοποιήσουμε αυτή τη γνώμη. Παρόλο που είναι φανερό και φαίνεται, δηλαδή πραγματικά θα ήταν εκπληκτικό να κάνουμε κάτι τέτοιο, το είδατε κι εσείς, αλλά δυστυχώς από πλευράς regulatory και αυτό ακόμη δεν μπορούμε να το κάνουμε χωρίς να το αποδείξουμε. Και για να αποδείξουμε negative results είναι ακόμη πιο δύσκολο από ότι positive results. Γιατί το είδος, το studies που θα χρειαστεί να κάνεις είναι πολύ μεγαλύτερα. Παρ' όλα αυτά, εκείνο που έχω δει μερικές φορές είναι ότι θα κάνουν το standard of care, αυτό που θέλουν να κάνουν, αλλά ξέροντας ότι κάτι δεν δούλεψε στο microcancer, πολύ πιο γρήγορα θα αποφασίσουν να κάνουν κάτι, να το αλλάξουν κάπου αλλού. Είναι δηλαδή πιο προετοιμασμένοι να σταματήσουν. Είναι πολλές φορές. Έχετε δίκιο, αυτό είναι πολύ σημαντικό.
Και μπορεί, αν μπορώ να κάνω μια συνέχεια, μπορεί να γίνει και outside of the patient, δηλαδή evolutionary, ας πούμε, πριν. Μπορεί κάποιος να προβλέψει τα βήματα μετά. Γιατί αυτό θα μπορούσε ενδεχομένως να γίνει σε κάθε ασθενή στην αρχή της μεταστατικής νόσου και να έχουμε έστω μια αλληλουχία.
Αυτό θα ήταν ακόμη πιο ενδιαφέρον. Αυτά τα 3D microcancers που έχουμε εμείς είναι περίπου σε 10 μέρες. Τελειώνει το πείραμα, 10 με 2 εβδομάδες. Και δεν είναι δηλαδή cell line. Προσπαθούμε να αποφύγουμε τα cell lines γιατί θέλουμε να είναι όσο πιο κοντά γίνεται στην πραγματικότητα. Για να μπορέσεις να κάνεις αυτά τα evolutionary experiments πρέπει να το κάνεις cell line και είναι δηλαδή ας πούμε τελείως διαφορετικό. Ο Πάνος πιστεύει ότι μπορεί να τα κάνει. Αλλά δεν είναι τότε πια για το... Ακόμη περισσότερο δηλαδή είναι δύσκολο να πείσουμε ότι αυτό πραγματικά γίνεται στον ασθενή.
Να ρωτήσω κάτι κι εγώ. Εντυπωσιακή ομιλία, ευχαριστούμε. Το ερώτημα είναι τι κάνει το Chromoplexic καλύτερο. Είναι ότι μπορείτε και αναγνωρίζετε ευκολότερα τους συνδυασμούς σε κάθε άρρωστο, ή γιατί παραδείγματος χάρη στην οριμότητά τους όλα τα sequencing project θα βρίσκουν deletions και amplifications γονιδίων. Δηλαδή είναι προφανώς όσο φτηνότερο γίνεται το whole genome sequencing, τόσο ευκολότερα θα γίνονται και όλα αυτά τα πράγματα, αλλά μπορεί να γίνει και πιο εύκολα και γρήγορα με άλλη μεθοδολογία. Οι συνδυασμοί δεν θα πρέπει να είναι άπειροι. Όπως είπατε, τα deletion του MHC class 1 και λοιπά είναι προβλέψιμα. Τα amplifications των key γονιδίων είναι προβλέψιμα. Υπάρχουν shortcuts ή είναι ανάγκη να κάνει κανένας whole genome sequencing?
Νομίζω στο σημείο που είμαστε αυτή τη στιγμή πρέπει πρώτα να κάνουμε, δυστυχώς πρέπει να περάσουμε το εμπόδιο του whole genome μέχρι να δούμε πραγματικά όλα αυτά που συμβαίνουν, να δούμε δηλαδή τα biomarkers και μετά να πάμε σε panels. Ο τρόπος που έγινε είναι επειδή οι τεχνολογίες ήταν ακριβές και πολύ δύσκολη πληροφορία, όλοι κάνανε single genes, double genes, small panels, then bigger panels. Αλλά έτσι όπως το βλέπουμε εμείς, είναι καλύτερα να τα δούμε όλα για αρκετούς ασθενείς, 1.000, 2.000. Και όταν αρχίζουμε να βλέπουμε patterns, τότε να κατεβάσουμε την πληροφορία. Πρέπει να περάσουμε αυτό εδώ πέρα το information barrier.
Ναι, δηλαδή στην οριμότητα του βλέπετε ότι όταν έχουμε καλούς κατάλογους από χιλιάδες, ίσως χιλιάδων, ότι θα μπορέσουμε να έχουμε shortcuts που να μην χρειάζεται αναγκαστικά whole genome sequencing, ή θα είναι γρηγορότερη μεθοδολογία, να πηγαίνει πιο εστιασμένα.
Ναι, έτσι πιστεύω, έτσι ελπίζω, για να μπορούμε να το κάνουμε πιο φτηνά και να μην χρειάζεται να κάνουμε όλο αυτό το functional genomics. Αλλά μαθαίνουμε, και αυτό εδώ πέρα το critical point θα πάρει καιρό, γιατί ακόμη από κάθε ασθενή μαθαίνουμε πάρα πολλά πράγματα. Δεν έχουμε φτάσει στο σημείο δηλαδή όπου η πληροφορία πια έχει γίνει saturate και ξέρουμε τι να κάνουμε.
Πόσο καιρό?
Κοιτάξτε.
Δηλαδή, τι πιστεύετε? Καλύτερο best case, worst case scenario? Λίγο βιαστική το γελάει ο κύριος.
Λίγο βιαστική.
Πολύ βιαστική. Απλά μια εκτίμηση.
Η εφαρμογή είναι τώρα. Τώρα αυτή τη στιγμή το κάνουμε στο MAYO Clinic. Δηλαδή έχουμε ασθενείς που μπαίνουν κανονικά στο πρωτόκολλο και όπως είδατε μερικές φορές. Η εφαρμογή είναι εδώ και έχει φτάσει αρκετά κοντά στην κλινική. Υπάρχει ένα Precision Medicine Clinic, το οποίο θα χρησιμοποιεί αυτό το Xvivo. Γιατί την πληροφορία που έχουμε αυτή τη στιγμή, όπως είπα, την περίπου είναι 30-40%, έχουμε τη γνώση να κάνουμε διάφορα πράγματα. Και έχουν βγει και αρκετά καλά drugs τελευταία, όπως είναι το Mutated για το KRAS ή ξέρω εγώ. Υπάρχουν δηλαδή πολλά πράγματα που μπορούμε να κάνουμε τώρα και τα κάνουμε. Αλλά όσο πιο complex γίνεται ο καρκίνος, τόσο πιο combinatorial θα πρέπει να κάνεις. Αυτό είναι που δεν ξέρουμε πολύ καλά. Πώς να κάνουμε καλό combinatorial treatments. Η γνώση αυτή για να γίνει σωστά θα πάρει κάποια χρόνια, αλλά είναι ένα iterative process. Κάνουμε ό,τι μπορούμε τώρα και μαθαίνουμε. Και όσο προχωράει θα γινόμαστε καλύτεροι και καλύτεροι με τα functional genomics. Δηλαδή εκεί το βλέπω εγώ.
Άλλη μια ερώτηση. Τα hypermutated genomes πόσο πιο δύσκολο είναι να μπουν στο χάρτη σας από τα genomes που δεν έχουν πολλά mutations?
Είναι ενδιαφέροντα τα hypermutated, γιατί κάθε φορά που έχουμε ένα τέτοιο δεν ξέρουμε τι να κάνουμε, γιατί όλα τα γονίδια έχουν χτυπηθεί. Αλλά το καλό είναι τουλάχιστον στο serous endometrial, τα περισσότερα είναι poly και αυτά τα poly είναι εύκολα, δηλαδή γίνονται πάρα πολύ εύκολα γίνονται cured με το surgery. Επίσης, το immunotherapy θα δουλεύει καλύτερα σε αυτά τα hypermutated. Αναλόγως τι είναι το hypermutation. Δηλαδή, πιστεύω ότι το immunotherapy θα δουλεύει καλά σε πολύ, αλλά μπορεί να μην δουλεύει καλά σε TMZ-related, σε drug-related hypermutation, όπου το hypermutation μπορεί να είναι πολύ heterogeneous.
Ή μπορεί να υπάρξει συνδυασμός. Γι' αυτό ήθελα να φέρω την κουβέντα στο immunotherapy, που στα hypermutated θα πρέπει να είναι μεγάλη βοήθεια.
Ναι, ναι. Σίγουρα και τα PARP inhibitor, όλα αυτά που χτυπάνε γενικά, ας πούμε, πιο γενικά, θα πρέπει να συνδυαστούν με τα targeted treatments με τον σωστό τρόπο. Αλλά και αυτό, για να το βρούμε, χρειαζόμαστε functional genomics. Αυτό προσπαθώ να εξηγήσω, ότι χρειαζόμαστε όλη αυτή τη μηχανή για να φτάσουμε εκεί, και κάθε ασθενής είναι διαφορετικός. Μην το λέτε, δεν έχουν έρθει στην Ελλάδα. Εγώ νομίζω, και γι' αυτό το μιλάω πολύ στην Ελλάδα, γιατί θέλω να τα φέρουμε αυτά τα πράγματα στην Ελλάδα. Υπάρχει τόσο πολύ complexity, που μόνο οι Έλληνες θα μπορέσουν να τα λύσουν. Οπότε νομίζω ότι είναι καλή ιδέα στο κάτω-κάτω έξω περισσότερο Έλληνες τα κάνουν.
Έχει να κάνει με το γονιδίωμα, δηλαδή το ιδιαίτερο.
Ακριβώς. Είναι νομίζω αυτό που κάνει τον Αριστοτέλη επιτέλους να είναι ευτυχισμένος.
Τέλεια.
Ζένια, έχουμε ξεφύγει λίγο από τον χρόνο.
Ναι, τι να κάνουμε. Έχουμε ξεφύγει εντελώς με αυτά που ακούμε.
Είναι καταπληκτική η κουβέντα μας.
Καλή συνέχεια διακοπών. Ευχαριστούμε πολύ.
Πραγματικά πολύ ενδιαφέροντα όλα αυτά, και ευχαριστώ τους διευθυντές για τις ερωτήσεις, γιατί θα έδειχνα στην καλύτερη περίπτωση η μάθειά μου σε σχέση με το αντικείμενο το οποίο αναλύθηκε μόλις πριν.